The overall goal of this procedure is to demonstrate the acquisition, detection, and analysis of local IP3-mediated calcium signals in intact mammalian cells using camera-based imaging. This is accomplished by first culturing the cells onto the glass bottom cover dishes and loading them with the cell membrane permeable forms of the fluorescent calcium indicator Cal520 and caged IP3. The second step is to position the cover glass onto the stage of an inverted microscope and illuminate the sample with 488 nanometer light to excite the calcium-sensitive fluorescent dye Cal520. Next, local calcium signals are induced by exposing the cells to a brief pulse of UV light. This photoliberates IP3 from a caged precursor such that it can now bind to the inositol trisphosphate receptor and mediate calcium release from the endoplasmic reticulum. The final step is to import the image stack into the custom-written algorithm to initiate the automated analysis of the recorded data. Ultimately, the algorithm rapidly localizes sites of calcium release with sub-pixel resolution, allows user review of data, and outputs time sequences of fluorescent ratio signals together with amplitude and kinetic data in an Excel-compatible format.
This approach allows for the imaging of hundreds of channels or events simultaneously, but yields vastly more data than traditional approaches. And the massive amounts of data generated render manual processing, visual identification, and analysis impractical.